Hello, friends. In today's session, we are going to talk about the human immunodeficiency virus in a nutshell in a very brief video. So to help me in this video put up the content, I have been helped by Dr. Shreya Singh, who's an assistant professor in microbiology, Dr. Neha Kumari, an assistant professor in pathology, Dr. Dhrivendra Lal, who is an assistant professor in community medicine, and we are at the Dr. P.R. Ambedkar State Institute of Medical Sciences, or in AIIMS, Mohali. So very briefly, talking about HIV, it's an enveloped retrovirus. It was believed earlier that the flow of information in biological systems is unidirectional from DNA to RNA. However, with the identification or discovery of the retrovirus, it was found for the first time that the flow of information is happening from the RNA reverse to the DNA. And this was made possible by the presence of a special enzyme called the reverse transcriptase enzyme in the HIV virus. So it is important to remember these facts, that there is this special enzyme, the reverse transcriptase enzyme, and the flow of information is from RNA to DNA. There are three important genes in the HIV. These are the GAG, GAG, POL, POL, and ENV, ENV. The GAG gene is responsible for coding for proteins towards the core and the shell, while the POL gene codes for the virus enzymes, and the ENV gene codes for the glycoproteins as part of the envelope or the membrane. The diagnosis of the HIV is dependent upon serological tests which look for antigens or antibodies, the viral RNA or the nucleic acid tests, and the culture of the virus, which is the most confirmatory test from body fluids or secretions or blood. The earliest possible diagnosis after an exposure to HIV infection or an infected individual or an infected fluid or a body fluid is at 10 days, and this is possible by nucleic acid tests. The antibody-based tests may take up to 90 days for detection of antibodies. The HIV targets CD4 plus T cells in the body, or these are also called the T helper cells. The life cycle of an HIV virus is in three important stages. The first is an infection where the virus attaches to the host cell, the integration of the provirus into the host DNA, and the replication of the virus to create more copies of itself. And finally, the third stage is the release of the virus from the cell, which causes the lysis or the death of the host cell. It is important to remember the routes of transmission of this virus. Please remember that no one is immune, and all body fluids can transmit infection, although in different degrees of transmissibility, where blood has got the highest grade of transmission. So the different routes of transmission which are important are those of unprotected sexual intercourse, sharing of infected needles, possibly during drug abuse, blood transfusions, and vertical transmission, wherein a mother could transmit the virus infection to her child through the breastfeeding or even during the process of childbirth during a normal vaginal delivery. So this in brief was a few facts about the HIV virus, and I hope this was useful to you. Thank you for stopping by.